Let's check. Hello everyone. A very good evening to all of you. So today is the week 12 problem solving session, and this will be the last problem solving session for this biochemistry course. And I hope you guys have learned and get prepared for your upcoming exam. So in this session, I'll be taking some questions uh, about the last week, and then I'll be taking some questions of from whole over the course, all the recaps. Okay, so let's see what's the first question we have. The first question we have, okay, what products of glucose oxidation are essential for oxidative phosphorylation? So, oxidation from the glucose oxidation, whatever products are released, which of them are essential for oxidative phosphorylation? So, is it acetylcoenzyme, pyruvate, NADH and FADH2, NDPH and ADP? What's the correct answer here? Any one of you?
Pyruvates.
Pyruvate. So, pyruvate is the last product of this oxidation, but I don't know whether it important for oxidative phosphorylation or not. Any other opinion on that? Okay, so it is NADH and FADH2. So, like in the electron transport system, the electron transport chain is present in the inner mitochondrial membrane. It comes to several electron transport in series, which facilitate the movement of electron from one carrier to another in redox reaction. The NADH and FADH2 produced during the citric Krebs cycles get oxidized. It results in the formation of proton gradient across the membrane, and which stimulate the formation of ATP by oxidative phosphorylation during cellular respiration. So here, the end product of cycles are CO2, NADH, FADH2, and ATP and water. So these many are the end product from the CO2, NADH, FADH2, ATP, and water. These are the end product from the this Krebs cycles. And the reactant of oxidative phosphorylation, it is say that NADH, FADH2, O2, ADP, and P. These are the reactant. So here the option is NADH and FADH2 is correct. In oxidative phosphorylation, this NADH and FADH2 produced during this Krebs cycle get oxidized. Uh, okay, so we can see the Krebs cycles here. So it is a common pathway for complete oxidation of carbohydrates, protein, lipids, as they as they are metabolized to acetyl coenzyme A or other intermediates of the cycle. The acetyl coenzyme A produced enter the this trioxylic acid cycle or citric acid cycle. Glucose is fully oxidized in this process. The acetyl coenzyme combined with 4 carbon compound oxaloacetate to form 6 citrate. In this process, two molecules of CO2 are released and oxaloacetate is recycled. Energy is stored in ATP and other high energy compounds like NADH and FADH2. So this NADH is the end product, FADH2 is the end product of this Krebs cycle. CO2 is the end product. Fine. So next question we have the combination of cofactors involved in the conversion of pyruvate to acetyl coenzyme. It is biotin, FAD or TPP, biotin NAD plus and FAD, NAD plus biotin and TPP, pyridoxal phosphate FAD and lipoic acid. What are the combination of cofactors involved in conversion of pyruvate to acetyl coenzyme? So if you see the cup cycle here you have a pyruvate. It got converted to acetyl coenzyme. So it is asking what are the cofactors are involved in this particular process from here to here. Is it biotin FAT-TPP, biotin NAD plus FAD, NAD plus biotin and TPP, pyridoxal phosphate FAD, or lipoic acid? What is the correct option over here? Anyone? Let me check whether I'm connected to all of you or not. Okay, connected. Yes, Arun.
Option C, sir.
Option C. NAD plus biotin and TPP. It is biotin, FAT, and TPP. Pyruvate to acetyl coenzyme conversion utilizes coenzyme A and NAD plus. Oxidized from nicotinamide adenine nucleotide as a cofactor. Thus, TPP, lipoic acid, and NAD plus are combination of cofactor involved in the pyruvate to acetyl okay, coenzyme conversion. So TPP, NAD plus. Okay, this is a contradictory answer. So yes, option C is correct, right? NAD plus, biotin, and TPP are involved. So during the oxidative phosphorylation of proton motif, force that generated by electron transport is used to create a pore in the inner mitochondrial membrane, generate the substrate ADP and PI for ATP synthase, induce the conformational change in ATP synthase, oxidize NADH to NAD+. What?
Option A, sir.
Create a pore in the inner mitochondrial membrane. Okay, option C is correct. It induces a
Yes.
conformational change in ATP synthase. Okay. So it induces conformational change in ATP synthase. So conversion of 1 mole of pyruvate to 3 mole of CO2 via pyruvate dehydrogenase and citric acid cycle also yield how many mole of NADH, how many mole of FADH2 and how many mole of ATP or GTP. So then we need to see the cycle, right? Uh, do you guys remember cycle?
Option C, sir.
Option C. Option C. 4 molecule of NADH, 1 molecule of FADH2 and 1 molecule of ATP. Correct. Okay, so you can see like curvature of Pyraway, 3 mole of CO2. You can see the Krebs cycle right here. I'll show you. This is Pyraway, right? So it generates 3 moles of CO2. 1 mole of CO2, 2 mole of CO2, 3 mole of CO2, right? And then generate 1, 2, 3, 4. 4 NADH and 1 FADH2, 1 ATP, right? So option C is correct. Which of the following is not associated with the oxidation of substrate by citric acid cycle? This pyridine-nucleotide oxidation, CO2 production, flavin reduction, lipoic acid present in some of the enzymes system.
Option C, sir. Flavin reduction.
Flavin reduction. Option A. Which of the following is not associated in the oxidation of substrate by citric acid cycle? So this pyridine-nucleotide oxidation. That is not associated with the curve cycle. So pyridine-nucleotide oxidation is not associated here. Okay. Which of the following is not an intermediate citric acid cycle? Acetyl coenzyme, citrate, oxaloacetate, succinyl coenzyme. Third thing, which one of these are not the intermediate?
Oxaloacetate.
Hmm?
Oxaloacetate.
Oxaloacetate. Okay, but oxaloacetate now. I think acetyl coenzyme. It's not intermediate citric acid cycle. Acetic acid cycle starts afterwards, so it is not the intermediate of the citric this cycle. That's why acetyl coenzyme is not intermediate of TCA cycle. TCA cycle starts when acetyl coenzyme bind to the oxaloacetate, forming citrate. So coenzyme A is released during the this step. So both acetyl coenzyme and oxaloacetate are not the intermediate TCA cycle. So these both are not the intermediate TCA cycle. Okay, you can see here. So this acetyl coenzyme bind to this oxaloacetate.
Yes,
This
sir.
then it generates the citrate and then it get converted to acetate. Then it's alpha ketoglutarate. Then it goes to succinyl coenzyme, the succinate, then fumarate, and then mylate, and then again oxaloacetate. So now next question we have conversion of 1 mole of acetyl coenzyme A to 2 mole of CO2 and coenzyme A while citric acid cycle result in the net production of 1 mole of citrate, 1 mole of FADH2, 1 mole of NADH, 1 mole of oxaloacetate. Which option is correct here?
One mole of FD,
Hmm?
one mole of FDH2.
Tell me again.
One mole of
1 mole of
FADH2.
FADH2. Yes.
Yeah.
So let's see. This is still consume 2 mole of CO2 and consume 1 mole of FADS2 will be generated in this Krebs cycle. Fine. Okay, 2 mole of CO2 produced in the first turn of the citric acid cycle have their origin in the carboxylic and methylene carbons of oxaloacetate, carboxyl group of acetate and the carboxyl group oxaloacetate, carboxyl group of acetate and the keto group of oxaloacetate, two carboxyl group derived from oxaloacetate. So which option do you think here?
Two carboxyl groups derived from oxaloacetate.
Hmm?
Two carboxyl groups derived from oxaloacetate.
Two
Option D.
carboxyl group derived from oxaloacetate. Correct. So option D is correct here. As you can see here, option D is correct. So this is oxaloacetate. Two carboxyl group derived from one carboxyl group, another carboxyl group. So this will generate this two CO2. And we can see it here. And see this oxaloacetate will convert to citrate and isocitrate. Then it will release one CO2. It will run release one alpha ketoglutarate will release on the CO2. Okay, next question we have here. Formation of one molecule of glucose from pyruvate requires 4 ATP, 2 GTP, 2 NADH, 3 ATP, 2 GTP, 2 NADH, 4 ATP, 1 GTP, 2 NADH, 2 ATP, 2 GTP, 2 NA, 2 NADH. Yes.
Option C. 4 ATP, 1 GTP, and 2 NADH.
Hmm?
4 ATP, 1 GTP, and 2 NADH.
4 ATP, 1 GTP, 2 NADH. Okay. Option A is
Is it
correct.
correct?
4 ATP, 2 GDP, and 2 NADH is required from the formation of one glucose from pyruvates. So you can see this option A is correct here. Let me take this. The synthesis of one molecule of glucose from two molecules of pyruvate requires 4 ATP, 2 GDP molecules as well as two molecules of NADH. The net equation is 2 pyruvate plus 2 NADH, 4 ATP, 2 GTP, 6 molecule of water, 2 S+N. It will generate this glucose 2 NAD plus 4 ADP, 2 GDP, and 6 phosphate. 4 ATP equivalents are needed to overcome the thermodynamic barrier to the formation of two molecules of energy-rich compound PEEP from two molecules of pyruvate. The glycolysis of conversion of PEP to pyruvate is a metabolically irreversible reaction catalyzed by PK. So you can see here that the complete cycle is given. This pyruvate is converted to oxaloacetate by pyruvate carboxylase, and here one ATP is converted to ATP. Again, PEP carboxykinase convert this oxaloacetate to PEP. One GTP convert to GDP, and then is phosphoglycerate. This again this ATP converted to ATP, it will form one 3-piece phosphoglycerate. This NAD plus is converted to NADH. This again it will convert to fructose 1,6 biphosphate, generate this 1 phosphate, fructose 6 phosphate, and then glucose 6 phosphate, and then again it will release this glucose 6 phosphate. Phosphatase will release 1 phosphate, it will deliver glucose. So this 2 ATP and 2 plus 2, 4 ATP plus 2 GDP, right, is required. Okay, so next question we have, which charger will convert solar energy into energy for the carbohydrate, into the energy of a carbohydrate? Chloroplast, Golgi apparatus, mitochondria, nucleolus. Which organelle will
Option
answer
C,
that? Which one?
mitochondria. Option C.
Mitochondria. It is asking for the solar energy conversion to carbohydrate. So it is asking basically to what plants that will be chloroplast. Okay. Here,
But chloroplast must be found in plants.
yeah, yeah.
It was one of the organelle in the plant. Chloroplast was one of the organelles in plants.
Oh, let me check one more thing. Yeah, it is for chloroplast found in plants.
It was one of the organelles in plants.
These diagnosis in plants.
Yeah, it's one of the organelle, specific organelle.
Yeah,
Okay, okay. So it was not a pigment.
yeah, no, no. These are organisms in the plants.
Okay, okay. Okay, sir. Then chlorophyll was one of the pigment.
Yeah, chlorophyll is the pigment.
Yeah, is it all right? Sir? Okay.
Hmm. Okay, so should we move on? So let's moving on to the next question. This glycolysis lead to the production of something and two molecule of ATP. In absence of oxidation, fermentation leads to the production of something else. Glycolysis plus secret cycle can convert this carbon of glucose to something, storing the energy as ATP and something. Electrons are transformed from oxygen to water. So, yes, fill in the blanks. Which option is correct?
Option B was the correct answer.
Option B. Pyruvate, lactic acid, CO2, NADH, FDS2. Okay, let's fill here. Let's see how does it feel, right? Let's see how does it feel. Glycolysis lead to the production of pyruvate. Okay, this is option B, right? Okay, let's say fill here. Pyruvate. Okay, and two molecular ATP. In the absence of oxygen, fermentation lead to the production of lactic acid. Okay, lactic acid. Glycolysis plus the citric acid carbon cycle can convert the carbon of glucose to CO2, storing the energy as ATP. Okay, here again NADH and FADH2 as storing the energy as ATP, NADH and FADH2. FADH2. Okay, so option B is correct, right? So you can see here this glucose first get converted to glucose 6 phosphate, and then again this glucose 6 phosphate get converted to fructose 6 phosphate by using this phosphoglucose isomerase, and then again this phosphofructose kinase converts 1 ATP to ATP by converting this to fructose 1,6 bisphosphate. Again that converts this aldolase convert this to GLAP, and then 1,3 bisphosphoglycerate. Then again phosphoglycerate kinase convert this to ADP to ATP and generate 3 phosphoglycerate, and phosphoglycerate mutation convert this phosphoglycerate to phosphoglycerate, and then enolase convert this enol pyruvate, and this pyruvate kinase convert to pyruvate. Okay, so option B is correct here. You're right. Fine.
Yes.
Yeah. Next question we have: citric acid cycle in eukaryotic cell takes place within the mitochondria, nucleus, cell membrane, cytoplasm.
Cell membranes.
Cell membrane. Okay, so it takes place within the mitochondria, not in the cell membrane. Okay, so this is takes place within the mitochondria. Next question we have: all of the above vitamins except one participate in TCA cycle. This is so all of the above vitamins, all of the following vitamins except one participate in TCA cycle. So it is asking which of all of which from the four which does not participate in TCA cycle. This pentosalic acid, lipoic acid, folic acid, this riboflavin. Which of
Option B, sir, lipoic acid.
hmm?
Lipoic acid.
Lipoic acid. Option C is correct, folic acid. Folic acid does not uh, contribute. Lipoic acid does contribute, right? As a cofactor. So this pantothenic acid is needed for the synthesis of coenzyme A, without which upcycle beta oxidation and other metabolism pathway will not work. So this pantothenic acid is required. So you can see here. Glucose, fatty acid, acetyl glucose plus fatty acid, it will form acetyl coenzyme. Glucose plus fatty acid, it will form acetyl coenzyme. Then on this oxaloacetate, it will it will form this citrate, and then isocitrate, it will get converted to isocitrate to alpha ketoglutarate, alpha ketoglutarate to succinyl coenzyme, then succinyl coenzyme to succinate to fumarate, and fumarate to mylate, and again this mylate to oxaloacetate. Okay, so that's short. Another question we have: which of the following factors is required for the conversion of succinate to fumarate in citric acid cycle? Just from last slide. ATP, biotin, FAD, and Na plus. Which one? Q.
Flavin adenine dinucleotide.
Hmm?
Flavin adenine dinucleotide.
ATP.
Options FAD. Flavin adenine dinucleotide. Option C.
So option C, FAD. So if you see from the last slide, the question is asking succinate
Yeah,
to fumarate. So FAD, right?
yeah,
Yeah, you can see here. This FAD is required to convert this succinate to fumarate. Correct?
yes, yes, sir.
So next question we have: which of the following cofactors is required for the conversion of succinate to fumarate in the? Okay, this we have answered already, right? FAD is required. Okay. Next question we have: this glucose labeled with 14C in C3 and C4 is completely converted to acetylcoenzyme by glycolysis and pyruvate dehydrogenase complex. What percentage of acetylcoenzyme molecule formed with the label with 14C? In which position of the acetyl moiety will the carbon 14C label be found? Yes. What do you think the probable answer could be?
Option B was the correct answer.
Option
Well, the
B.
percentage of the acetylcholine will
100%
be
of
labeled by
acetylcoenzyme will be
C.
labeled with C2. Okay, I think there will be no label only found in acetylcoenzyme molecules because all the carbons will not be coming from the glucose. It will be coming from the this pyruvate dehydrogenase complex. So no label will be found in the acetylcoenzyme. Okay, so option D is correct here. So now we'll be taking again few questions from all the whole syllabus, and then it will be just a quick recap. Fine. So next question we have: which of the
Yes.
following single letter codes is present in the structure below? Find out. I think this is some mistake. This will be O. So tell me what the single letter code for this peptide. So what is the N-terminus?
Option A was the correct answer.
Hmm?
Option A, ASGYVD.
ASGFVE. ASGYVD. Okay, let's. This is correspond to This will correspond to A, right? And this will correspond to yes. And this will correspond to G. This will correspond to okay, not from here. Y. This will correspond to here again. Pose missing V. This will correspond to D. So option A is correct, right? Okay. Next question we have amino acid can be classified according to polarity and side chain group type. Okay. From the option below, choose the one which contains amino acid with only aromatic side chain. Which is correct with the only aromatic side chain?
Option D was the correct answer.
Okay.
Phenylalanine, tryptophan, and tyrosine.
Okay. Phenylalanine, tryptophan, and tyrosine.
Tyrosine.
What is the aromatic in phenylalanine?
Yeah, these three amino acids are aromatic amino acids.
Okay. No. What is the side chain in tryptophan? It is indole modified.
So,
So next we have a buffer is prepared with 1 molar HA and 1 molar NaA. pK of HA is 4.87. The pH of the solution H. What will be the answer?
7.40.
7 point
7.40.
7.40. How do you calculate?
For my guess.
Okay, it is your guess. Okay, so here if you see pH is what? pH is based on the concentration of H plus, right? Right? pH
Yes, sir.
pH is based on the
1 to
concentration.
6.9.
HA will give what? HA plus plus A minus, and Na will give Na plus plus A minus.
A minus.
So if you mix these two both, then also the concentration of H plus will not change, right? Isn't it? Hmm? right now. So if the concentration of this A will not change on the basis of this Na or H, so the pH will be same as 4.87. So that's what the correct answer is A. Because this pH is equal to pKa plus log A minus divided by concentration of Ha. So if this value will be 0, right? This will consider to be used, and pH will be pKa. So then the pH of the solution will be 4.87. Okay. So next question is we have here gamma carboxyglutamate or phosphoserine or phosphotyrosine are few enzymatic modification of amino acid that form after protein biosynthesis. They are very important component of cell signaling pathways. Okay, so such modification commonly known as translational modification, post-translational modification, or post-transcriptional modification, post-translational modification, transcriptional modification. Yes.
Option A is translation modification.
No, these are post-translational modification. So it is saying that right after protein synthesis. So translation will be till process. It will be till protein synthesis, right? Till synthesis.
Yeah,
It will be till synthesis, right? And and it is protein biosynthesis. After protein biosynthesis processes occur, it's called post-translational modification. So translational and post-translational. So this is post-translational modification. Fine.
okay, sir.
So next, our students synthesize two novel amino acid A and B. Okay, whose structure are given. The hypothetical hydrophobicity is calculated according to the Doolittle scale, which set represent the correct hypothetical value. A, B, C, D. Yes. So hypothetical amino acid have a positive value and this okay hydrophobic amino acid have a positive value and hydrophilic amino acid have a negative value according to this Cardinal scale. So hydrophobic, which one will be hydrophobic? Will this be hydrophobic or this will be hydrophobic? Yes, tell me. So first one is A is hydrophobic, right? So if it is hydrophobic, it should have a positive value. B is a hydrophilic. So if it is hydrophilic, right? And this is hydrophobic. So it will have a positive value. So A has a positive value in these two, and negative B has a negative value in this. So option A is correct, right? Which are the following nomenclature for the tripeptide whose structure is shown? Which option is correct?
EDK, option B.
K, K, D,
EK, EDK,
K, D, E, right?
option
Okay,
B, EDK.
E, D, K. Option B. Okay, which one is
Yeah,
E option? Which one is E amino acid? This one? This one is E amino acid? E, D, and then you mean that this is a D and this is a K. So, however, EDK will be the correct answer if it is drawn properly, right? This should be drawn in which way? It should be drawn. Let me try this for you. It should be drawn in S2, 1, 2, COH, right? CO, NH, again COH, then CO, NH, again 3, 1, 2, 3, 4, then NH3 plus, S3 plus, here COO minus. If it have been drawn in this way, right, then it will be E D K. But the drawing is not correct. From right, understood. The structure does not represent a peptide correctly. So conventionally, the first left amino acid has the N terminus free, and the last amino acid has the C terminus free. But you are right. Another question we have. The question says, in an alpha helix, hydrogen bonds are between the side chain of a polar amino acids and CO of NH of the backbone. Between CO of a chain to NH of another chain that runs side by side. between the NH of I and CO of I plus 4 in the single chain, between CO and I and NH of I plus 4 in the single chain. So which option is correct? Option D is correct. Between CO of I and NH of I plus 4 in alpha helix. So if we draw alpha helix, so if this is if it's CO and this is NH, then this will be I plus 4. And then this H will form this hydrogen bonding. This is I. Okay, next question we have. From the diagram provided, find the correct statements. A represent an alpha helix at the C terminus. B represent an beta turn between antiparallel strands. C represent an adjacent parallel beta strand. A represents an alpha helix at the N terminus. So which statements are correct here? Tell. Any of you?
A represents an alpha
Option A,
helix at
first and
the
second.
C terminus.
Okay. Option B is correct. Second and fourth. So
Okay.
B represent the beta turn between antiparallel strand. Yes, this B is a beta. These two are antiparallel strand, right? And fourth A represent alpha helix at the N terminus. So alpha helix at the N terminus. It is not at the C terminus, it is the N terminus. So this is not correct. And the C represent adjacent parallel beta strand. It's not adjacent, right? Antiparallel. Okay, next question we have. For the portion of peptide shown below, which of the following statements true? Means what does it stand for? The angle called.
Option B was the correct answer.
Which one?
Y phi angle.
Y is a
It's a
phi
phi angle.
and Z is a psi angle, right?
Yeah. Yeah.
This is a phi and this is a psi.
PSI angle.
Yeah. Next question we have: which of the following best describe the assumption made in steady state kinetics? Kinetic analysis. The concentration of S is decreasing. The concentration of ES is constant. The total amount of enzyme decreases. So steady state approximation is E plus S gives to formation of ES that lead to the formation of product plus enzyme free. So concentration of S decreases. Um, Concentration of ES is constant. So here the assumption made is this concentration of ES is constant in the steady state approximation. Okay, so option C is correct. Fine.
Yes, sir.
Okay. The number of substrate molecule converted to product in a given unit of time by a single enzyme molecule at saturation is referred to as dissociation constant, half saturation constant, Michaelis Menten number, turnover number. Yes. Which one? Tell now. So it's a turnover number, right?
Option C is correct answer.
Option D is correct, right? It is a turnover number.
Okay, sir.
Yeah. So next question we have, which among the following type of enzyme can add water? Isomerase, ligase, transferase, lyase. So option. So here lyase add water, ammonia or carbon dioxide across double bond or remove this element from to form the double bond. So its option is lyase. Okay. So next question we have, enzyme X exhibits maximum activity at pH 6.9. X shows a fairly sharp decrease in its activity when the pH goes much lower than 6.4. One likely interpretation of this pH activity is that a glutamic residue on the enzyme is involved in the catalytic reaction. A histidine residue on the enzyme is involved in the catalytic reaction. The enzyme has a metallic cofactor. A reaction relies on specific acid-base catalysis. So, what information can be withdrawn from here? So if the reaction is being much lower than 6.4, that means it will have a histidine residue. Histidine residue, if it has, then if you decrease the pH below to 6.4, this histidine will be protonated, then the reactivity will be shut down, right? It will be decreased. So option B is correct. Okay. Next we have an enzyme catalyze a reaction at a velocity of 20 micromole per minute. When the concentration of substrate S is 0.01 molar. The Km for this substrate is 1.1 into 10 to the power minus 5 molar. Assuming that maximum conditions are followed, what will be the reaction velocity be when the concentration of S is these two? A 10 to the power minus 5 molar and B is 10 to the power minus 6 molar.
Option B was the correct answer.
B is the correct answer.
1.8 mu mole per
At least
minute.
calculate na.
Yeah, yeah, it's my guess.
Oh, this much guess.
Yeah,
So see, the question will be V naught is equal to this much guess is not good, bro. Km into substrate concentration. So the velocity of 20 micromole per minute is essentially Vmax because it is measured at substrate concentration is way higher than Km. Once the substrate concentration is way higher than Km, if the concentration of S is way higher than Km, no, then V naught is equal to Vmax. Km plus. So that is V naught is equal to half Vmax, right? So once the substrate concentration is given, S is equal to 10 to the power minus 5. S is given 10 to the power 5, and this Km is 10 to the power 5. So you can calculate it from the Maclaurin mentioned equation. So option A will be correct, 10 micromole per minute and at 1.8 micromole per minute. So it will become 10 micromole per minute and 1.8 micromole per minute. Okay. So let's see next question. Based on the requirement of activation energy, enzymes can be defined as those which do not require activation energy, lower the requirement of activation energy, increase the requirement of activation energy, do not change the requirement of activation energy. Make a guess here. Make a guess here. So it will lower the requirement of activation energy. The enzymes they will lower the requirement of activation energy, right? Next question: which among the following can act as a choline esterase inhibitor? This choline, insecticides, phenyl benzoate, ethyl acetate. And this is, let's see, the answer B is correct. Okay, so let's take next question again. This is the above conversion catalyzed by succinate dehydrogenase. So dehydrogenation reaction. What will be the product if succinate dehydrogenase was to react with the molecule X? So it is asking if this is succinate. It reacts this is catalyzed by succinate dehydrogenase. It dehydrogenates these two bonds, these two hydrogens. If you see here, these two hydrogens, and then it will form this double bond. This double bond. So what will be the reaction if this succinate dehydrogenase will have to react with this X molecule? Yes, tell me.
reaction does not occur.
Reaction does not occur. Okay, correct answer. Why do you think so? because enzymes are very specific towards substrate, right?
Yes.
Yeah, enzymes are very specific towards substrate, so reaction will not occur.
That lock and key model.
Yeah. So here we have enzyme, substrate will bind, inhibitor will come, it will bind to the here inhibitor, substrate, binding this cycle. The given diagram shows a certain inhibitory effect where E stands for enzyme, S substrate stands for substrate, I stands for inhibitor. For the depicted enzyme substrate interaction shown, the inhibitor I can bind either E or ES. So inhibitor can bind direct enzyme or this enzyme substrate complex. So the identify the inhibitor process. Uncompetitive inhibitor, competitive inhibition, non-competitive inhibition. The reaction is not an inhibition reaction. So what's right here?
Option B, competitive inhibition.
Competitive inhibition. Option C is non-competitive inhibition because in this inhibitor can bind both. It is not binding to the substrate binding position, right? It is not binding to here. Either substrate is there or not. Inhibitor will go bind to this position. That is not substrate binding position. This position. Here enzyme inhibitor binds, right? So that means there is no competition between substrate and inhibitor. So option C is correct. So option C is correct. Depicted enzyme substrate mechanism has a structural specificity. Okay. Identify the specific species unique to this kind of inhibition. Ternary complex formation of binary complex, or formation of a quaternary complex, formation of polar intermediate. So option A is a ternary complex formation. Enzyme plus substrate plus inhibitor. It's a ternary complex. Fine. So option A is correct. Proteases can are involved in digestion and diet proteins, digestion of diet proteins, maintaining turnover numbers, regulating certain enzyme activities, cell signaling. Which option is correct here? First, second, third,
Option
fourth.
A was correct.
Hmm?
Yeah, yes, sir. Option B.
Option B. First, second, third, fourth, all are correct. Yes. Right. So option B is correct. So this all option was correct. Okay. Let's see. The catalytic triad of most serine proteases contains aspartic, histidine, and serine acid. Which of the following is not true? The aspartic acid is used in acid catalyzed attack on the peptide bond. The histidine residue is responsible for substrate specification of trypsin. The serine residue is used in nucleophilic attack on the peptide bond. The aspartic residue is responsible for the substrate specificity of trypsin. So select which is not true. Yeah, option C. First, second, and fourth. These are not true. The serine residue has one OS that is able to act as a nucleophile attacking the carbon of a successful peptide bond of the substrate, right? So this is not true. The residue catalyze attack on the peptide bond. Aspartic do not use the acid attack. Histidine residue does not is not responsible for specificity of the trypsin. and the spotting residue is responsible for the substrate specific substrate. No, so option C is correct. On the molecule, we have given the molecule's own coordinates with the metal ion M2 plus for their functioning in the human blood. When X coordinates with M, this structure is referred to as Y. M and Y are, yes, till now. So M is the molecule's own coordinate with the metal M2 plus with for the function in the human blood. So M is Fe, Y is a heme,
Option
right?
yes.
The answer B is correct. So this is a porphyrin. Porphyrin, right? Next question, we have a certain peptide fragment is treated with a demand reagent CNBR, cyanogen bromide, carboxypeptidase separately. Treatment with the demand reagent yields B. Okay. While treatment with cyanogen bromide yields VGA. Lactone and
Option yes,
urea
sir.
lactone and LPSR fragment. On the other hand, treatment with carboxypeptide yields R. So identify the peptide fragment.
Option E.
Which option?
Option E is
Option A.
option E.
E.
Yeah.
Okay, E option is not A, B, C, D only are there.
A E
A,
R G A M L P S V.
P, S, V. Option D is correct. CNBR hydrolyzed peptide bond at the C terminus of methionine residue. So CNBR hydrolyzed the C terminus of the methionine residue. So it will hydrolyze here. This reaction is used to reduce the size of polypeptide recombinant information sequencing. So option D is correct. VGA. In demand, it will degrade this N terminus. So N terminus is V. It one degrade N terminus is A, and it will degrade here this CNP this VGA lactone. So this remove this methionine, and then LPSR and R this carboxypeptide reduce this stop at C terminus. So this option D is correct. Okay. So next we have, which of the following product is responsible for yellow color observed in the Nithari test? Yes, option B is correct. So option B is the this combination of this complex is responsible for the yellow color. Okay, the given figure shows the schematic representation of various types of membrane proteins. Identify the types of protein labeled as X, Y, Z. So X, these X proteins, these are peripheral. Y are integral and Z are lipid anchored. This is peripheral and these are lipid anchored. Option A is correct. Which of the following act as a barrier to most water soluble substance? Phospholipid, hydrophilic heads, nuclease, deoxyribonucleic acid. So here the barrier for to most water soluble substance is phospholipids. Answer A. Which of the following option does not correspond to the pyrimidine bases found in RNA? Uracil and thymine, thymine and cytosine, adenine and thymine, cytosine and uracil. Which option is correct? Option D. First, second, and third. They do not does not correspond to the pyrimidine bases found in RNA. So only cytosine and uracil are found in RNA. Identify the structure of XYZ. XYZ. It's a nucleobase, nucleoside, nucleotide.
Option D of the correct answer.
Nucleotide, X is nucleotide, Y is nucleoside, Z is
No.
nucleobase. Okay. Option A is correct.
Option D.
See, this is adenine, right? This is nucleobase. This is nucleoside. This is nucleotide. Once phosphate is attached, this is nucleotide. So option A is correct here. Rate of hydrolysis is slower in DNA than RNA. This is because which region? The ribose sugar present in DNA in the deoxy form, but it is not so in RNA, or of the presence of different pyrimidine RNA compared to DNA, of the stacking of nitrogen bases of the phosphate in the backbone of RNA favors hydrolysis at a faster rate. What's the case?
Option C of the stacking of nitrogen bases.
So, this question we have taught right here only. So this ribose sugar present in DNA in the deoxy form, but it is in
Nitrogen
the
bases.
but not so in RNA. So if you see this basic difference, right? What is difference here? O minus. This is H in DNA. This is OH in Here you have connected base. Here you have a phosphate. Base, phosphate. So if you have OH here, now then OH this can form negative, it can go and it can hydrolyze these other nucleotides. That's why it is unstable. So this is DNA, this is RNA. Fine. And this question is most
Yes,
likely
sir.
to come. This is very pet question to us in this kind of exam. The overall rate of DNA regeneration is determined by the concentration of DNA, the average length of DNA segment, the complexity of DNA. First and second, third only, first, second, third, first only. Option three, first, second, and third. It is regeneration is determined by all three options, right?
Okay, sir.
The section is dehydrogenase. Identify AB state in the vitamin involved in the reaction. So this is what FAD, FADH2, riboflavin.
Option A was the correct answer.
Option E was the
FAD, flavin adenine
FAD,
dinucleotide.
FADH2, riboflavin. Right. So option A is correct answer. So the structure shows is adenosine triphosphate. It is an important versatile nuclear molecule that can donate the XYZ moieties. The name the Z moiety. What is the name of the Z moiety? Gamma phosphate, beta gamma phosphates, adenosyl group, or adenosyl group AMP. Option D,
Option
adenosyl
A
group
was the correct
AMP.
answer. Vidogama was the
Adenylyl group AMP is correct answer here. While eating cooked egg is highly nutritious, eating raw egg is advised against. Given that raw eggs contains avidin, that may be possible reason for this. Avidin binds tightly to riboflavin, making riboflavin less available for its enzymatic function. Avidin cannot be digested by the gut bacteria. Avidin binds tightly to folate, making folate less available for its enzymatic activity. Avidin binds tightly to biotin, making biotin less available for its enzymatic function. So it is avidin biotin binding is well known. So it is option D. So I think we should stop here, right? And the last slides are related to drug discovery, which I was thinking to discuss, but I think it's time to disperse. Thanks a lot for joining, and I hope you guys enjoyed your learning and hope to see you do well in your examination. Okay.